This is the last part of the microscope lab. I've gone into the pond tank and taken some fronds off of the plants. The fronds almost always have animals around them, and that's what we're gonna look for. And so I'm gonna put this on this microscope. Really tough to do that and keep it dry on the bottom, so I've gotta kind of mess with it a bit before I put it up here. Let's kill the lights. I meant to do that before. Okay, this is low power. Let me see what I've got. Lights, way too much light. Since these things tend to be semi-transparent, I need to close my iris diaphragm. And what I'm gonna do is work my way along the plant, because usually the bigger, more drawable critters are associated with the plant, so. There goes one swimming by. Very difficult, because what I can see through here and what you can see on the screen are not that similar. So I have to find stuff and then focus it for me, and then focus it for the screen. Turns out I need to move it somewhat. Okay, these are what are called cup ciliates. I had some of them out for some of the classification lab. You can see some movement in here. It would be nice if they were as focused on the screen as they are in my microscope. But they're a little cup shaped. They have little stalks that attach them to the plant. Okay, I'm gonna go look for something else that's maybe a little better than that. I'm gonna go back down to low power. There's a big old hydra.
Can you point to it? I think I've got it.
I'm getting there. I need to move it where it's on the screen right. Okay, hydra is attached to the plant here.
Hold
Don't
on.
block your own screen.
Okay.
Here's the body of the hydra. Here's the tentacles of the hydra. Here's little creatures that crawl around on the hydra that actually are single-celled protozoans that instead of having cilia to swim with, they fuse them together at the tip and they use them like little feet. So that's all you can really see moving. I'm a little surprised. Oh, there we go. The hydra's moving a little bit. Not entirely sure what's over here. I'll have to see what that is. It's moving, but I'm not entirely sure why. Okay, so that's another creature. There's a creature.
Saw something moving.
Yeah. Oh, he went underneath. That, I believe, was a baby flatworm. Oh, there he goes. Part of what this is training for, if you do it in the lab, is being able to focus and chase and focus and chase. Yikes. He had to go around an air bubble. And now I've lost him because I'm trying, there we are. There he goes. Fortunately, since you're on video, you can freeze frame these things when you need to draw them. When you're trying to draw them in real life, you have to kind of get a good image of them and then, oops. And then try to draw, and then you look back and they're gone. Okay, that's another critter. Let's see what else we can find. There's the hydra. There's a lot of plant here, so I've got to focus up and down through the depth of this drop of water. This is not something you would want to put a cover slip on to flatten it out, even though it would reduce the up and down that things can swim in. but it also reduces a lot of, your ability to kind of dive through looking for things that are moving. Okay, this is another type of Cup-ciliate.
Can you point to it?
You can see it here. If you watch it, it'll make a little jump after, there it goes.
Oh, yeah,
It's not doing a lot of moving, but it's doing a little bit of moving. And it is attached to the plant. It might actually be a flagellate. That might be a flagellum attaching it to the plant. I actually have not seen this particular creature before, so I'm not entirely sure what it is. I know it's a protozoan. There's two of them, but other than that, I can't, let's see, I'm trying to get it. That's about as good a focus as I can get on the screen. Okay, I'm moving on. There's a lot of algae in here, but algae doesn't move. There goes something zipping by. Yeah, unfortunately, when something gets to the edge of the water drop, the way that light veers through the water drop makes it difficult to keep them in focus. Also, what's in the middle of my field is not in the middle of the screen, so I have to find it and then look back up at the screen. Where's this little guy? Well, I don't think we're gonna get, now I'm off the drop entirely. There is a funny little guy, doing a lot of zipping around here. This is definitely a single-celled eukaryote. It's a protozoan. There he is.
I don't see him.
I don't know if you could even draw that. He's moving so fast.
Yeah.
There's the hydra again. Now the hydra's, moved back a little bit so you can get a better view of it. You can still see the creature crawling on it. I'm gonna put a little bit more light behind it. Just a little bit more. All those bumps on the tentacles have stinging cells on them. When I had one out for the classification lab, it had stung and swallowed a fairly large animal. That's why it looked big and blobby. Okay. Let's see if there's anything else. Usually there's insect larvae I'm lucky enough to catch. I don't think I got any this time. There's another one of those swimmy things. Very often you're looking at things that are just too small to get a really good look at. And they like to go places where you can't see them. Well, you've got a couple of choices of things to draw. I'm gonna keep looking and before I decide that I've run out of, I think I've gone all the way across all of these plants. But let me go way over here and see if there's, I may put more plants on here than I actually ran across. Nope. This is all the way at the other end, and those two things that I had before. Surprised there's no rotifers. Usually there are rotifers attached to these plants, but they haven't been this time. There's the hydra again. Okay, that's it. So what you're gonna do is draw one of the things that was moving under here to show, and you would be showing in the lab that you could actually do that. You could find it, you could do a reasonable drawing of something that isn't necessarily gonna be cooperative. But that's it for that part.